Hello, welcome to the Spermatology lab at the University of the Western Cape in Cape Town, South Africa. I'm going to show you today the ideal setup for a spermatology laboratory, but also with special reference to computer-aided sperm or semen analysis. And let's first look at the really very basic requirements. First of all, we need a good microscope. And there are a couple of very important points here, and that is that the microscope must have phase contrast microscopy for human sperm, positive phase contrast microscopy, and also the stage must be heated. And here we have the controller for heating the stage. This is crucial. You cannot just work with the stage at room temperature. Of course, a camera that feeds into the computer, and the images are captured here where it is analyzed. We will come to that detail later on. What is next required? Of course, in order to work with semen, you need to have a really good pipette set, when you do sperm concentration, you actually need to work with a positive displacement pipette. In this instance, there is no negative pressure created. Accordingly, you use these little pistons and you insert it into the pipette like this, and this piston pushes right through the tip, and now you can accurately take a semen sample and pipette it slowly, no air bubbles, no negative pressure, and onto the slide, which has been temperature heated, controlled, and of course inserted into your particular chamber very slowly. And what is good about this pipette is that it now displaces absolutely all the semen. No semen sticks to the sides. What else is required? Yes, further temperature control. For example, where do you heat your tubes, your slides beforehand? We have this special incubator, but any incubator that controls at 37 degrees is fine. We also have one here for heating up slides before the time. And then, of course, if you want to do long-term experiments where you need to control the pH and you work with a bicarbonate buffer, we're going to use a CO2 incubator like the particular one we have here. So you can see how much I emphasize temperature control. You cannot do without it. You must heat your pipette tips, you must heat your tubes, you must heat the slides. All of it must be at 37 degrees. Sperm is very sensitive to temperature, and there is no compromise to that. It doesn't help that you heat your slide at 37 degrees, but you use a tip which is still at room temperature. That creates a temperature shock, and it is not acceptable. While we are focusing near these temperature controllers, we also have very special centrifuges. Often it is important to wash your sperm and to slightly centrifuge. We have these that are that can do all the required speeds. And here we have a very special one which works between 4 degrees centigrade and about 50 degrees centigrade. In other words, if you do long-term centrifugation, it means that you can actually keep your sperm sample at that same temperature. Yes, what else is important? We spoke a lot about temperature control, but what about media? Yes, you need to warm up your media also long before the time, but you must take a lot of precaution to keep your media in a good, well temperature controlled refrigerator, and I will show you some of the more important media that we use. In this instance, I am going to show you a kind of general sperm wash that we use. There are many of these equivalent to human tubular fluid, for example. We also like to work with this medium, which is a D-MEM medium or a Hams F12 medium for animal sperm. And then, of course, we have this 40 and 80% gradient, which we use to separate sperm. And this is very useful if one wants to obtain a sperm pellet with a very high concentration of motile spermatozoa. So you can see all these solutions are kept in the fridge at 4 degrees centigrade, and only small amounts are aliquoted and heated up to 37 degrees. Again, very important to heat it up at least an hour before the time, and then only then use it. Okay, now that I've shown you the basic requirements for a Castle laboratory, which is indeed very simple, but one must be very thorough about it, we can go into the details of specifically computer-aided semen analysis. And let's start off with the details of the microscope. So we're going to concentrate here on all the components that are required really. So one needs a really good, solid compound microscope. In this instance I'm using a Nikon E50i microscope, and it is fitted with, first of all, a very good halogen source. It has a green filter, which is essential for positive phase and negative phase contrast microscopy. It has a phase contrast condenser, and then of course I've indicated the heated stage. And then before we of course do any measurements, we heat up the slide here also, and we fill the slide. And of course here we have a 10 times positive phase lens which is required. The next step is of course that you require a really good camera with a sufficiently fast frame rate. Commonly we work with frame rates between 25 frames per second for most human work, but also of course for hyperactivation studies of human sperm or sperm cervical mucus penetration, we need to of course work at least at 50 frames per second. There are a whole range of these fast frame rate cameras. They can go up to actually 100 frames per second. We have this specific little camera which can do only black and white, but it is a basilar camera that can do also 50 frames per second, but can even go up to 100 frames per second. In some instances, in some species, this is required. In addition, there are a whole new range of basilar cameras. There are also these scout cameras, and they are both ones that work with firewire, but also with 8 giga Ethernet. And so, of course, it's very easy to exchange these cameras. One just really undo this lid, and you take this camera off, put this one on, and exchange the cables, and there you are. And so it is very easy to exchange it should you require to work with a different camera or higher frame rates. The next step is that this signal, for example the motile sperm, is now fed into your computer. And again, here one must take a great deal of care. You cannot just use a computer with any settings. It's preferable to use a Hewlett-Packard computer with really lots of RAMs, very fast processors, and then make sure that if you work with Giga Ethernet that it has the proper Giga Ethernet card at the back. So that is the absolute important requirements for being able to start your work at all. Now that we've completed the setup, all the components of the computer-aided semen analysis, let us show you how we do it. First of all, we load the semen sample. And again, a positive displacement pipette is used. We inject the sperm carefully. We leave it there for approximately a period of 30 seconds. In this time, I have already selected the configuration that I require, in this instance human sperm. We're just going to do a short demonstration of how the motility component actually works. I go into the SCA program. For the time being, we will close the database just to show the demonstration of motility. We select the particular configuration we need for motility. We're going to analyze now. We focus sharply on the sperm. It's not a particularly good sample, which is typically what one gets in the clinical situation. I'm just optimizing the light a bit. And we analyze. Yes, and now the swimming sperm appears in different colors, all depending on what kind of swimming behavior there. For example, for fast sperm or for slow sperm, and we can check it under results. We can view the field. We can replay to check the accuracy. The yellow dots would be immotile sperm, the red would be fast progressive sperm, the green less progressive according to WHO4, or we can do WHO5, where it again changes the colors and group A and B together, and the yellow would be the immotile sperm. Here we can view the results, and there are many options like looking at the total motilities, looking at aspects such as hyperactivation, and looking at the kinematic parameters. We can finally make a report, or we can save this also as a mod file. Now that we've completed the motility, we're going to show you how to make a proper sperm smear. And Asanelli is going to take 10 microliters of the raw semen, the neat semen, and she's going to put it on the side of a so-called star frost slide. These are star frost slides. They electrically treated to adhere more sperm. She's going to put 10 microliter also on a duplicate slide of the star frost type slides. And now you can see how she's going to actually make the smear. And this is very crucial how you make it. She moves the slide in towards the drop. The drop doesn't touch the drop yet. Now it touches the drop at the back of the slide, and it drags the fluid, the semen, behind. So no sharp edge can cut the sperm. So she brings it quite from the top, brings it close to the drop of sperm. It fills the area behind the slide, and she completes the slide. At this moment in time, we will have to wait about 25 to 35 minutes for the slide to dry properly so that we can stain it. While we are waiting, we can just indicate that we use a stain called Sperm Blue. It's a new formulation that stains within 45 seconds. It contains both the fixative and the staining component in one solution. And one just needs to insert the slides very slowly in there and leave it for about 45 seconds, and then gently put it in the stilt water again for about another 6 seconds and dry it. And that is all. We will show the procedure just now. Okay, Asanelli now puts the slides into the Sperm Blue, which is at room temperature, and we leave it there for exactly 45 seconds. And while we're not going to do it now and just demonstrate the technique, after 45 seconds she will remove the slides. You can remove the slides, drip dry them on filter paper, and now you can insert it in distilled water for 1, 2, 3, 4, 5, 6 seconds. Slowly pull them out and Yes. And you can lean it against there, and that is fine. We can normally just take off most of the stain also at the tip, and also clean the back of the slide. This is really important. Lots of people do not take care to clean the back of the slide. Thank you. And you can see that is a very nicely stained slide. This one, maybe there was too much sperm, too much seminal plasma proteins there, and maybe it's not a good one to use at all. Perhaps stain another one. We're going to show you how to do a proper morphology analysis from sperm blue stained slides. Okay, we're just starting the programs again, and this time just a very brief review of sperm morphology. We again select the correct configuration for human, and we can start directly analyzing as we have already set up the configuration nicely. We're focusing there on sperm stained with sperm blue. We analyze. And we can analyze just one more field. This would obviously be a micro, I would imagine. Yes. And let's look at the results. And you can see here is indicated as a normal sperm. It has green around it, that as abnormal. And we can actually see the features by viewing this field. We can see here why this is abnormal. It has an abnormal size, and this can be seen with the sperm information. The width is too wide, 1.52 micrometers, so it is a little bit too fat, the spermatozoon, compared, of course, to the one here in green. So the important thing here, just showing this field again, is to show how well the sperm are actually stained, how well the SCA recognizes the acrosome, the rest of the head, the mid-piece, the implantation, and then of course classifying it accurately. And what helps a lot with the sperm blue technique is that the background is nice and clear, and this is so important in image analysis. It is not the intention of this to show you all the modules of the sperm class analyzer and CASA analysis, but rather to show you the typical setup in a lab. And here you can see if you want to do fluorescence, for example vitality staining, you need again a good microscope, but then with of course epifluorescence attachment. And in this instance we have of course a super high pressure mercury lamp with which we drive the lamp which sits in this housing, and here we can select of course our filters, the different kind of filters required, and it must be done in absolute darkness of course, and that is a typical fluorescent image that you see on the screen. And so this is the basic setup again for fluorescence microscopy. Now one can do a lot of other fluorescence techniques with spermatozoa, which has nothing to do with vitality but shows other facets. For example, the acrosome reaction, and one can then have a very nice image of a fluorescent sperm, of fluorescent acrosomes, and one can capture that in the sperm class analyzer's cell counter and analyze it there semi-quantitatively. So this brings us to the end of this first program that we wanted to show you, and we really hope you enjoy your conference. I want to thank Asanele, I want to thank Sia for being such great assistants. Thank you. Bye.